Hey again, guys. Make sure you guys are on page 3 of your notes. It's kind of sad looking at this because I really wish I could show you some microscope slides about this process and it would make it a little more interesting, but it's really hard to do not being there. But nevertheless, here we are. So this whole page is about the stages a cell goes through. If you were to take your cells and put them under a microscope, you would see that most of them look the same. A lot of them look like they're just hanging out, and that's represented in this diagram right here by something called interphase. And so if the cell is just hanging out, not really doing too much, and I say that, but there is an important thing that can be happening here. That's interphase. But that's what most of our cells are doing. They're just chilling out. So this whole thing is something that happens in eukaryotic cells. And if you remember, eukaryotic cells are plants and animals, are found in plants and animals. And if you remember from the other day, all of our regular body cells, which are not sperm and egg, are called somatic cells. So remember, the other day we talked about how all of our normal body cells, like muscle and nerve and white blood cells, are called somatic. Sperm and egg are sex cells. It's kind of weird that they wouldn't they're not calling sperm and egg our body cells, but it's because their purpose isn't just to make up our body and to have our body function. Their purpose is different. It's for reproduction. So somatic is just body cells. But all of our somatic cells go through what they call the cell cycle. And the cell cycle has 3 main stages. The interphase stage, which I said is mostly just hanging out, not doing too much. The next one is when the cell actually divides, which you learned the other day is called mitosis. If we're making sperm or egg, it's called meiosis or meiosis. And then finally, this kind of gets lumped in with this sometimes, but this is called cytokinesis, and that's just when the cell actually splits into 2. Nothing fancy. But let's start with this big stage here. Make sure you've written that if it's not already in your notes. I think that's in your notes though. But here's the start. If this is the start, the first stage is G1. Yes, I skipped one. The next stage is S. The next stage is G2. So as you can see, the after interphase would be mitosis and cytokinesis. But this is definitely the longest stage. Cells spend most of their time in interphase. But let's see what happens here. One, this cell's going to grow. It just kind of grows a little bit, make sure it has enough nutrients, and the goal is to get here. So we're just going to make sure that we have enough nutrients and we're growing the cell, and then we have to make sure DNA is copied. Sometimes they say it's copied, sometimes they say it's replicated, sometimes they say it's synthesized. So DNA synthesis, that is why it's called S phase. And then G2, they generally just say that there's more growth and more preparation for division. So very general. terms there. There are some cells though that will exit this whole cycle, because a cell can go through this, divide, go through it again, divide again, go through it again, divide again. Our skin cells are a good example of that. But some cells, they don't really divide anymore once you have enough of them. They go out here into what they call the G0 phase. Make sure you write that here and then in your notes. An example of that are your nerve cells. The second you have all of your nerve cells, if you damage them, you're not just gonna make more, sadly. It's just done. This is a picture of what interphase looks like, and if you remember the term from the other day, is this chromatin form of DNA or is this chromosome? So hopefully you remember this is chromatin. If it's just a jumbled mess of DNA, it's chromatin. Okay, so out of all these three, this is probably going to be the most important one, because this is where you replicate your DNA. And I talked a little bit about this the other day, how if you have 46 chromosomes and you divide that cell in half, you're going to get 23 in each one. And then let's say each of those divide in half. That would kind of give you an uneven number. But then that would be cut in half and you would keep depleting your DNA. So we don't want that to happen. We don't want to get rid of our DNA. So instead, we have 46 chromosomes to start with, and then we have 92 when the DNA gets replicated, so that when we finally split the cell, we actually have the 46 in each. Okay, so let's actually talk about mitosis then. Let's assume that we went through all of interphase, and now we're finally here at mitosis when the cell actually divides. A lot of times they call this division of the nucleus, because you'll see in these stages of mitosis, we're mostly just talking about the DNA, how it coils up, how it aligns in the middle, and then how it finally divides. So we're mostly just talking about the nucleus and DNA. The DNA and the nucleus. And it has 4 stages: prophase, metaphase, anaphase, telophase. And you can remember that by just remembering PMAT. P-M-A-T, PMAT. You will not be tested over like knowing these names specifically. If you are, they're already going to be on the test. So as long as you remember P-M-A-T, PMAT, you can put them in order, hopefully. So it sounds kind of foreign right now, but it should be easy to put them in order if you just remember PMAT. So here's prophase. Looks like this. What happens is the chromatin, remember the jumbled mess, begins to coil up into chromosomes. We're going to have to get some of these chromosomes over here, some of them over here. So in order to do that, we had to take them out of the nucleus. This whole thing is the cell, so we don't even see the nucleus. If it'd probably actually be a good idea if you show the nucleus, just that it's disappearing. This actually, I want you to do. Please show the nucleus disappearing. There's another thing happening here, and that's that there are centrioles, sometimes called centrosomes, and they move to opposite ends, and what they're going to do, you can see them here. They're going to shoot out these ropes to grab a hold of the chromosomes to pull them to each side. And those ropes are called spindle fibers. So centrioles, just kind of like these little things that shoot out ropes to grab the chromosomes to pull them to each side of the cell. Metaphase, you're going to see those centrioles, the spindle fibers, and those chromosomes have moved to the middle. So as I talk about this, you know, you can get into a lot of detail in biology, and the more detailed you get, the more you kind of ask yourself, like, why would I need to know that? And they actually take more and more out of the state requirements for what you need to know about mitosis. So really there are just a couple things that you need to know about each stage. This one, that you see chromosomes. This one, that the chromosomes go to the middle. And I've starred the important things. Anaphase is when they're going to be pulled away. So you can kind of see that they've been split right down the middle, and half of that chromosome has gone this way, half of that chromosome has gone this way. So chromosomes are pulled apart, and those sides, the sister chromatids, are pulled towards opposite sides. So the other day, you don't have to draw this, but you saw a picture of a chromosome and you labeled each one of these sister chromatids. And you can kind of see how those sisters split. Sadly enough, the sisters had to split down the middle. So one sister went this way, one sister went that way. Finally, the last stage is now we have our chromosomes on each side of our cell, and they're going to kind of unwind again because we don't need them in the chromosome form. We don't need to organize them anymore. So they relax and they just chill out again. So now they're at opposite ends. Spindle fibers, those ropes disappear, and chromosomes unwind back into the jumbled mess of chromatin. And we're going to need that nucleus again now, so the nuclear membrane starts to reform. So let's kind of go through and show where everything is. Remember, the jumbled mess is chromatin. Here we see our new membrane, new nuclear membrane. Each one of these is called a chromatid. Here you can see the chromosomes are in the middle. And even though they formed up here, they don't really show a picture of them. The spindle fibers, remember, just think of them as ropes. Are all of these lines. I don't know why I made that line so big, but all of these ropes that shoot out are the spindle fibers. Centrioles are just, they're kind of hard to explain, but they're just these molecules on each side of the cell that kind of look like this, and they just shoot out the ropes. This is where we first saw our chromosomes, right here. And here you can see the nuclear membrane disappearing. So, like I said, you know, the main part about cell division is that we're dividing a cell so that we can grow and heal. If we do too much of it, that's where you get cancer. Cancer is when you have uncontrolled cell growth and they're just dividing out of control. And you'll learn a little bit more of that soon. But the details of the steps, you know, that's not really too important. Just that you know, like, the basics. Okay, to divide a cell, we're going to have chromosomes. They're going to have to go to the middle so that they divide perfectly evenly. And in the end, we get a cell that has an even amount of chromosomes. So one last thing, you can kind of see in that picture, our cell still hasn't split. So we've gone through all of mitosis and the cell still hasn't split. So they give that another stage name. Cytokinesis. That's when the cytoplasm, all of the liquid and all the organelles actually splits into two. So they kind of show it more here. Like, obviously it hasn't finished, but eventually it will, and you'll get two new cells. So in animal cells, that pinching is called a cleavage furrow. In a plant cell, instead of pinching in, the wall can't pinch in because it has a hard wall on the outside. So instead, vesicles just start to form in the middle until it separates. So this will just keep going and going until the whole thing splits into two. And those pieces are coming from that Golgi apparatus that we talked about. And this is called a cell plate. So that's how a cell divides. We'll talk more about why that's interesting later, and how you know, if a problem happens with this, then major problems can happen in your body. So hope you guys are having a good day.